Okay, so we are looking at Chapter 3, Water Relations. We're still in Module 1, looking at the individual and its environment. Last time we talked in Chapter 2 about light, and this time we are talking about water relations and energy. So where did I leave off? Is looking at water potential, flaccid, turgid were a couple of the no. So looking at water potential, we can see how the environment can play a role, or how the tree itself can alter its parameters to be able to modulate how they move water around in its tissues. Okay, so let me kind of put this into a nutshell. Think about it this way. Plants feed themselves. Light is extremely important. That was chapter 2, right? Light is fundamentally important to autotrophic organisms, because if you're not a chemotroph, if you're a phototroph, you're feeding yourself using light energy. So that's extremely important for a plant biologist, a plant ecologist, a plant physiologist to understand how plants do that. And we talk about, well, how come you're doing so well here, but this guy isn't? What's the difference here? And we go in and take a deeper look at the ecology and the physiology of what's going on between those two things, so we can understand better how communities are structured and how adaptations work or don't. Okay, so we looked at light. We talk about a lot of different aspects of light. Including how it behaves as a particle and how it behaves as a wave. Okay, we talked about spectrum, the wavelengths that are useful to plants. Two primarily are useful to plants: red light and blue light. Those two wavelengths activate photosynthesis. Okay, and the other thing we talked about was far red light and phytochrome, this on-off switch. We talked a little bit about wavelengths of light and how those wavelengths are attenuated as full spectrum light moves down through the light canopy. Do you remember my diagrams on the board? Kind of getting you to make sure you go back and take a look at your previous notes. So as I review, you guys are going, yeah, I got that in my notes. I got that. I understand. Oh, wait a minute. I had a question about that. Okay, kind of thing. So reviewing your notes. So as light moves down through the plant canopy, it moves through matter, through leaves, through branches, and all that stuff. And some of the wavelengths of light are going to get changed out, either being absorbed, reflected, or transmitted. The most light that's transmitted through upper layer canopy of plants down to the understory is far red light, pretty much uninhibited. However, red light, because it's so important in photosynthesis, sucked up. As red light is moving down through the photosynthetic leaves, we're using it, we're absorbing it, so very little of it actually makes it down below. So numerator red, denominator far red, small number for red light underneath the canopy, right? Because most of it's gone, it got absorbed, but most of the far red light gets pushed right on down through, transmitted down on through. So we're asking questions. My dissertation asked this question. So we've got all these plants in the forest understory. What kind of light environment is it dealing with, and what role does phytochrome play in its ability to adapt and survive and reproduce in that light environment, very low light environment? So we are interested in those kinds of things. As a plant ecologist, I ask those questions in my dissertation. Okay, what role does phytochrome play? All right, so I already told you guys that it's the meta file that you are going to be using from now on. That's where all materials are going to be posted. Meta merged your original file with Luke's original file, so now Luke is in the classroom parameters, and I can just post things once and everybody gets them. Okay? All right. So far as chapter 2, then we looked into some of the adaptations for photosynthesis, C3 and compared C3 to C4 in CAM, understanding how those things worked. Did quite a bit of work on that. Anybody have questions about that? One of the things that I stressed to you was just simply the logistics of how to study in an upper level class. And I gave you two examples in chapter 2. I gave you the C3, C4, and CAM table of comparisons. I told you to compare them. Your book didn't. So what would you do in future? You look for those things, for kind of pulling things together so you can kind of tell the difference between things and annotate and compare, right? So these are skills and tools that you're going to try to bring to the game in future chapters. We also did that in later things, distributions, for example, we did that. And then we actually went in and looked at where do we find these things? Looking at the distribution map and kind of pinpointed some patterns and things and asked questions about four corners. I got to tell you, I did this with Luke when he comes to my office on Wednesdays to meet, and I showed him that diagram with New Mexico, Arizona, right in there, and I said, "Do you know what that area is right there?" And he said, "Oh, that's Four Corners." Yes, that's Four Corners. So he recognized it right away. Kudos to Luke. So he saw that. And then we moved from chapter 2 into chapter 3, adapting to life on land. And I kept talking to you about how plants, because they have to feed themselves using light, they do it by grabbing CO2 from the atmosphere, bringing it in through the stomates into the leaves where it gets fixed into organic molecules, carbon and hydrogen rich molecules like sugars, okay, and carbohydrates. Now, open the door, CO2 has to come in, open the door, water gets out. It's a massive trade-off. And the way the plant reduces loss of energy, loss of work that they've already accomplished, they do that through C4 and CAM photosynthesis to kind of minimize the trade-off of water loss and maximize the uptake and hanging on to and using CO2. But then there's also the answer of, well, if they lose water, and they will, because the stomate's open to let CO2 in, how do they get water into their system, and how do they move it around, and how do they adapt to living on land? We talked a little bit, a reminder to you guys about how plants move from an aquatic existence to adapting to live out of water, and that took some major advancements. The major thread that we talked about in botany was going through the plants that live on land that are very simplistic. Come out of water, no longer have water, but on land, the bryophytes, the mosses and the ferns are intimately tied to wet environments. We move from that to larger plants a little bit farther away, still tied to water, but a little bit farther away from water, and now they're just a little taller. Well, if you're not washed in water, how do you get water up here? Vascular tissues came into play, okay? So we're looking at ferns and their allies, early plants that are becoming adapted to living life on land. So bryophytes, mosses, and liverworts, ferns and allies, and then going even farther away from, bless you, consistent water resources are our gymnosperms. Loblolly, we've got loblolly that we're going to be studying here. And loblolly, the pines and gymnosperms in general are extremely well adapted to living on land. There are naked seed producers, and then we move into our most highly evolved group of plant organisms, and those would be our flowering plants, our angiosperms. They have the most complex vascular tissue system of our plants that are categorized in the plant kingdom. Okay, that was our quick review of where we came back, where we're landing in talking about how water relations and energy balance play out in looking at plant ecology. And that's the topic of chapter 3. Agreed? What happened to getting that guy up and loaded. So when we got into chapter 3, we were talking about some terms such as turgid, flaccid, and we looked at what happens when you put a cell into different solutions. If you have chapter 3 open, it's the blue beakers with the cell that goes into the tube. I believe that's right about where we left off, talking about turgid versus flaccid. Okay, so these are the figures that I gave you in the handout. They are not in your textbook. So in case you're looking for that, that's from a previous lecture in an earlier class. Okay. One other thing that I mentioned that you're going to see in your book. There it is. Oh, goodness, that machine is so slow. I will do my best to try to expedite this in future. This is just terrible. Sorry about this. We just talked about how we believe as evolutionary science is showing us that plants move from algae, living in an aquatic environment, became adapted to life on land, bryophytes to ferns to gymnosperms to angiosperms. We can kind of draw an evolutionary line between these advances and changes and adaptations over time to life on land. And I pointed out to you that when you go back to an aquatic system and study the water plants that exist today, we believe that algae to land, adapted to land, moved back to water, and adapted to living to water. So they kind of went full circle back into an aquatic system. As they did, we can show evidence that these guys devolved some of the characteristics that make them habitable on land. Cuticle helps protect water loss. Aquatic plants lack cuticles, but they have all the other different functioning parts, except they want water to move freely into their very thin cell tissue layers, you know, instead of having to work at moving water up from the soil and about the plant. So something that we'll be talking about and looking at that. So, getting there? Yay! Oh, yeah, it looks like my Wi-Fi stalled out. Sorry guys, try this again. Here we go. Okay. So, yeah. Here we go. All right, so where we left off was looking at water potential and this diagram discussion of what happens when you put a living cell into a high solution concentration